Hello and welcome to my video tutorial on the second chapter, which is sexual reproduction in flowering plants. And I must say this chapter is important both in terms of your future career if you wish to pursue in botany, as well as from the CBSE examination point of view. And I want you all to pay extra attention to the lessons and make sure that you understand the concepts thoroughly. Now for today, I'm going to be taking up the structure of anther and Microsporogenesis. And here are the objectives of this session. 1. To identify the essential and accessory walls in a flower. 2. To distinguish the male and female reproductive structures. 3. To describe the structure of anther with a labeled diagram. 4. To list the 4 walls of anther and their functions. And 5. To explain Microsporogenesis and microspores. And I expect each one of you to achieve these learning objectives by the end of this session. Now to start with, let us recall the reproductive orbit. Yes, you are right. We all know that they are the flowers. And I assume that everyone loves flower. I do love them. So flowers not only add colors and beauty to our life, but they also are vital for the reproduction in plants. Now let us look at the flower as a biologist. Biologically speaking, a typical flower has 4 walls in it. And they are the outer wall, which is the wall of calyx. And inside of the calyx there is corolla. And next to the corolla we can be expecting the wall of andrecium. And internally there is the gynoecium. So these are the 4 walls in a flower. Okay, so if we have to understand these 4 walls in a flower, let me use this pre-drawn let's look up here to my pre-drawn flower, and this is an LS of a flower. LS, which means to say it is a longitudinal section. Right? So, in a flower, as I said, we have 4 walls, right? And the outermost wall is calyx. Now, what is this outermost wall calyx? Calyx is actually a collection of sepals. And all of these walls of the flower basically arise from a kind of a bulbous, swollen region, which is at the terminal end of the pedicel. Pedicel is the stalk of a flower. So at the terminal end of pedicel, there is a swollen region known as thalamus, or it can be even called as receptacle. So from the thalamus, we have the 4 different walls of the flower arising out of it. And in the outermost region, there is the wall of calyx. Now calyx is, as I said, a collection of sepals. And sepals basically are these green leaf-like structures. Alright, and they are functionally important to a flower when a flower is in the bud stage. So it can be providing protection to a flower when it's in the bud stage, or it can be even helping in you can say holding or the supporting of the floral parts when the flower blooms. And in the next wall there is corolla. Now what is this corolla? Corolla is actually a collection of all these brightly colored petals, right? So all of the petals together collectively are known as corolla. And corolla, as we understand, most of the corolla are bright, or they are very colorful. And the reason why they have such bright color is in order to attract the pollinators so that they can be helping in the process of pollination. And because of which, we say the outer two whorls, that is the whorl of calyx and corolla, since they are not directly involved in reproduction. So these whorls are considered to be the accessory whorl. Alright, so accessory whorl in the sense they are the supporting whorls, whereas the inner two whorls, right, in a typical bisexual flower, we can be expecting a flower to have these two components, and the inner whorl, which is the whorl of andrecium. So andrecium is actually a collective unit of the male reproductive structures. That is to say, these are the collection of stamens. Alright, whereas gynecium is the female reproductive unit, and this is basically composed of one, or you can say many, of pistil or carpels. So andrecium is composed of stamens and gynecium is composed of the carpel or pistil. Okay, now if you look into the parts in a stamen. So what is present in the male reproductive unit, which is a stamen? So stamen basically has two main components. They are at the tip of the stamen, we have a structure which is known as anther, and then beneath the anther we have a long stalk which is called as filament. Whereas in the gynecium or in a carpel, we expect three of the major structures here. They are, at the tip we have the stigma, followed by a long slender thin tube which is called as the style, and at the base of the carpel we have a swollen region, and that swollen region is attaching to the thalamus, and this is called as ovary, and within the ovary we can be expecting the ovules. Right? Now in order to get a kind of a better understanding of the different parts of the flower, I will show you a kind of a very quick dissection of a flower. Right? So what I have here is, we have this flower. This is a very common flower. It's called as the thura. So I'm going to be having a dissection of this flower so as to show you with the internal parts. Okay, so here is the thura flower. This is a huge flower, so it's easy to see the different parts. So here is the pericyle, then the green leafy, the sepals, which forms a fused structure here, and then the brightly colored petals. Okay, so I'll do a quick dissection of this so that we can see the inner parts. Now first thing, we will cut open the sepal. So I'll cut open the sepal and pull it out from the base. Okay, so here is the fused sepal. Next we have the corolla or the petals, right? So we will cut open the petal and see the inside of this flower. Okay, so here is, so we'll cut it from the base. Okay, so here is the inner part of the petal, and inside now you can also see these elongated structures, right? And these structures that I'm holding here, so these are the stamens, right? And as we know, stamens have anther and filament. So here are the anthers, which looks very beautiful here, and I hope you can be seeing the bilobed nature of the anther as well. And then we have the long filamentous stalk, right, which goes deep into the flower and is then attaching to the thalamus. Okay, so I will just cut out this floral part. So we have the petals here along with the stamens, okay, with a long filament, right? And then finally what we are left here with is, we are left with the gynecium, which is the female reproductive unit. And in the gynecium we have the base, which is a bulbous structure. This is the ovary. Then it has a long tubular stalk, which is the style, and then at the tip of the style we have this head-like structure, a platform, right, which is called a stigma. And if I feel this, I can feel the stickiness of the stigma, because it has to collect all the pollens that are going to be deposited here. So this is the female reproductive gynecium. Okay, and if you want to be, as we know, inside the ovary we have ovules. So if you want to see the ovule inside, what you need to do is you have to be basically cutting open this with the help of a blade and we have to make a transverse cut. But the thing is, since we are not having a microscope, we will not be able to see right now. So inside the ovary, we have the ovules, and this finishes with our flower dissection of the datura. So we have the sepals, we have the petals, then we have the stamens, right? And here we have the gynoecium. Next, let's try to look at the structure of a stamen. So in a stamen, there are two main parts, the anther and filament. Now let me draw a simple diagram of a stamen. So here is a stamen, and this head part of the stamen is the bulbous head part of the stamen. So this is the anther, and the long stalk-like, thread-like structure, this is filament. Okay, and the filament helps in attaching or exposing the anther and attaching it to the floral part. Particularly here we have a swollen region at this end of the filament, and this is called as the proximal end of the filament. So the proximal end of the filament attaches to the thalamus, or it can be attaching to, in some of the flowers, it can also attach to the petals. Right? So these two forms the basic unit of a stamen. Now, if we need to look into the structure of an anther, so now we are to be focusing more on the structure of an anther. So if we look into the anther, we find that in most of the flowers, anthers are mostly bilobed structures. Okay, so as I have drawn here, you can see two of the lobes here in the anther. So because an anther usually has two lobes, an anther is said to be bilobed. So if you talk about structure of anther, the first feature that we can say is that an anthers are mostly, you can say, bilobed. So it is bilobed. Anthers are bilobed. What do we mean by bilobed? It consists of two lobes. And if we try to go into the internal structure of the anther, which means, I mean to say here, if we take a cross section of the anther, or if we cut the anther transversely, so we will be able to see not only the two lobes, but there are even inside the lobes, inside each of the lobes, there are compartments which are known as theca. So, and because of which anthers are not only bilobed, they are said to be dithecous. So, anthers are bilobed and dithecous. Alright, which means to say it has two theca. Theca is actually referring to a sac, or you can say a kind of a compartment inside the lobe. So each of the lobe of the anther has two theca, and because each lobe has two theca, it's called as dithecous. Alright, now to get a better idea, let's try to have a cross-sectional diagram of an anther, which means to say I'm going to be having the TS of an anther. Okay, we are going to be cutting the anther transversely. Okay, so what you see here is, if we take the transverse section of an anther, it seems to be having a tetragonal structure, right? So you can see here we have four of the sides here, And in each of these sites, okay, in each of these sites, we can be finding a structural circular sac-like units, right? And these units, which are carried inside the anther, are technically referred to as, so these are referred to as Microsporangium. Microsporangium. Alright, so an anther has two lobes, and each of the lobe, it has two theca, two of the compartments. Hence the anthers are bilobed and dithecous. And if we take a cross section of the anther, we are able to be finding a tetragonal structure consisting of 4 of the Microsporangium at the 4 corners. Right? And because of which we can also describe the anther as tetragonal, consisting of tetrasporangia. That is to say, they have 4 microsporangia. Alright, so I'm going to be writing these are tetragonal, alright, and tetrasporangiate. So why do we call this as tetrasporangiate? Tetra means 4 and sporangiate has to be referring to the microsporangia. So we have 4 of the microsporangia, right? Microsporangium is referring to one of the sporangia. Microsporangia is the plural of microsporangium. So tetrasporangiate is because it has 4 of the microsporangia, right? Now these microsporangia or microsporangia are enclosed inside 4 different wall layers of the anther. So an anther has 4 of the wall layers, which can be, or the cellular layers, which can be enclosing the microsporangia, right? And we are going to be learning even with the different 4 of the cellular layers which are present in an anther. Right? So this is a typical structure of an anther. Now, if we try to have a kind of a further understanding of the anther, this anther also has from these indentations. So we have a kind of depression here. So from these corners, from these edge, we have a structure which is known as the line of dehiscence. So the anther after maturation would be carrying many of the pollen grains, and the pollen grains needs to be released. So in order to release the pollen grains, mature pollen grains, anther would be dehiscing, which means to say it is going to be breaking open. And this dehiscence is occurring from the line here, right? So we call this as the line of dehiscence. To dehisce is to mean to break open, right? So the anther is on maturation, the anther is going to be opening from such lines here, right, which runs throughout the length of the anther. Okay, so this is a typical understanding about bilobed dithecous anther, right? Now what we are going to be doing is we are also going to be looking into, as I said, the 4 wall layers of the anther which envelops or surrounds are microsporangia. Let's look at the TS of a young anther. TS means to say here transverse section. So in a transverse section of an anther, as we discussed earlier, we can observe the tetragonal structure consisting of the 4 microsporangia, right? And because of which we call even the anther as tetrasporangiate. Now these Microsporangia are enclosed inside 4 wall layers, right? And the 4 wall layers I have depicted with different colors here. And the 2 lobes of the anther are connected together by a kind of a sterile tissue, and these are called as, so the tissue that you're seeing here, so this tissue which connects the 2 lobes of the anther together, they are called as connectives, right? Then if we go into the 4 wall layers, now which are these 4 wall layers? The 4 wall layers are, from the outer side we have a layer which is called as epidermis. So here I'm showing the layer with a red color. So the outermost layer is epidermis. Under the epidermis, it is followed by yet another layer which I have shown here in green color, and this layer is called as endothecium. So this is endo because it is beneath, it is inside of epidermis. So it's a layer which is called as endothecium. Then inside of the endothecium, there is yet another layer which I have shown here in blue color. So this layer is the 3rd layer called as middle layers. Middle layers. Alright, now we'll get into the detail in the next part. And then the innermost layer close to the Microsporangia, right? This is the layer which is called as tapetum. So these are the 4 wall layers which we find in an anther. So the anther has, from the outside, an epidermal layer followed by an endothecium, then with middle layers, and on the inside of the anther we have tapetum which connects to the Microsporangia. Right? Now this is a kind of an important figure which is commonly even asked in your CBSE board question paper. So we can be having an enlarged view of such a TS of an anther. So I'm going to be showing just a part of it because we cannot be drawing the whole of the figure. So what we do is we try to just take a part of this. So let's take a blowout of this region and draw it here with the different cell layers. Alright? So I'll be drawing the enlarged view of a Microsporangia with the 4 wall layers, right? In the enlarged view of a Microsporangium, what I've shown here is I've shown the 4 wall layers specifically. So in the outermost layer, we have the layer which is epidermis, and epidermis is basically a very thin layer, a very thin single layered cell, and these cells are more flatter in appearance, right? So we have the flat, thin layered single layer of epidermis. Then inside the epidermis, there are these second layer which are called as endothecium, and endothecium cells are usually more thicker. They have more of the thicker cell wall deposition, and these cells have a kind of a more elongated appearance. So they are more radially elongated, right, cells, and these cells are even single layered cell. So it is a single layer of endothecium which are radially elongated cells, and inside of the endothecium then we have multi-layered 3rd layer which is known as middle layers. Now, it is multi-layered and the cell have a kind of a more circular kind of appearance. So, we can be expecting 2 or 3 layers of such circular cells, right? Collectively known as middle layers. And on the inside, the innermost layer, which is again a single layered cell. And these layer are known as tapetum, And tapetum is a layer of a cell having a kind of a pyramidal shape, right? And they are in direct contact with the innermost cells of the Microsporangia. That is to say, these layers have a direct contact with the Microsporangia, right? And in a young anther, in the Microsporangia, right, they can be consisting of a compactly arranged homogeneous cells which are known as sporogenous tissue. So I've shown the sporogenous tissue here in brown coloration. Okay, so these are the sporogenous tissue in a young anther. And such sporogenous tissues would eventually be, you can say, maturing, right? As the anther matures, the sporogenous tissue then would be making up or transforming into MMC, or we can say they transforms into PMC. Okay, MMC, which is microspore mother cell, and PMC, which is the pollen mother cell. So such is the understanding about the internal structure of an anther when it is in the young stage, where the microsporangium containing sporogenous tissues are enclosed or surrounded by 4 of the different wall layers of the anther. Right? So the 4 wall layers are, let me go back to the 4 wall layers again. So we have on the outer, from the outside to the inside, we have outermost layer which is epidermis, the second layer which is endothecium, the third layer which are the middle layers, and the innermost layer which is tapetum. Right? Now if you look into the functional aspects of these layers, the outer 3 layers which are epidermis, endothecium and middle layers, they have a kind of a collective function of protecting the young anther, or you can say they can be protecting with the pollen grains or the microspores present inside. Okay, so they are protective, as well as these layers can be eventually later, as the pollen grains have matured and when they are ready to be released, these outer 3 layers would be helping even in anther dehiscence. So we said an anther would be having a line of dehiscence from which the anther would be breaking open, right? And for such kind of dehiscence, the 3 layers, that is the epidermis, endothecium and the middle layers, are very, very helpful. Then the innermost layer, which are the tapetum, tapetum being the layer direct in contact to the sporogenous tissues or the microspores or to the pollen grains. So this layer is mainly related with providing nourishment to the growing microspores or providing nourishment to the growing pollen grains. Right? And not only that, tapetum is also thought to be secreting an enzyme which is called as callase, and this enzyme is important for the, you can say, the development of pollen grains from the microspores, which we will come in our next section when we talk about microsporogenesis. Right? So functionally speaking, the innermost layer, tapetum, has a very significant role because they are essential basically for the development of the microspores as well as for the development of pollen grains. And if tapetum is not present in a kind of a proper, you can say, state, or in a proper tapetum has to be having a lot of cytoplasm. So tapetum cell is quite unique in terms of having a dense cytoplasm, as well as it can be even multinucleated. And because of such characteristic, tapetum is able to be providing nourishment to the sporogenous tissues as well as to the pollen grains. Now, let's try to understand what is an MMC or PMC. So as we said, the young anther would be having, inside of the tapetum, it would be having these cells which are called as sporogenous tissues, right? And from the sporogenous tissues, as the anther starts maturing, now they are going to be transforming and making MMC or PMC. And MMC stands for microspore mother cell. PMC stands for pollen mother cell. Now let's try to understand why these cells are named as such. Okay, so the cells are called as microspore mother cells because these cells, right, the MMC, which are there inside the Microsporangium, right, these cells which are diploid cells, because we know angiosperms are diploid plants, right? So their cells are going to be diploid cells. So the diploid cells of the MMC, that is microspore mother cell, would be dividing and giving rise to producing spores which are called as microspores. And because they give rise to microspores, they are known as microspore mother cells, cells which are able to produce microspores. So they become microspore mother cells. Now, why the name pollen mother cell to these cells as well? This is because the microspores would then further divide and differentiate and form into pollen grains. So from the microspores, gradually we are to be obtaining pollen grains, and because of which MMC, or it can be even referred to as PMC, because these cells finally would be transforming into pollen grains, right? Now the basic idea is how we have the formation of the microspores and how we have the formation of pollen grains taking place inside the Microsporangia, right? So to understand this, let me give first a kind of an overview of the process here. So MMC which are diploid would undergo a division and result in the formation of haploid microspores. Alright, so MMC undergoing cell division and producing daughter cells which are called as microspores, and these microspores are haploid spores. Okay, these are haploid male spores because they are produced inside the male reproductive organ. Then the haploid microspores would then further divide and then differentiate into haploid pollen grains. Okay, pollen grains are also haploid cells. So such is the basic understanding of the changes that are, or the divisions that are taking place during the process of formation of microspores and pollen grains. Now let's try to have a recollection of which type of the cell division can be resulting in the production of microspores from a diploid MMC, and which type of cell division would be producing a pollen grain from a haploid microspore to a haploid pollen grain. So can you guess the nature of the cell division? So if a cell starts from a diploid parent cell and then results in the formation of a haploid daughter cell, right? In this case, the daughter cells are the microspores. So such cell division has to be reductional cell division, because we find a reduction in the number of chromosome copy, right? And hence, here the nature of the cell division would be meiosis. So when the microspore mother cells, the diploid microspore mother cells divide meiotically, right, it produces haploid microspores. Now, these haploid microspores on further division and differentiation results in haploid pollen grains, right? And because there is no further change in the chromosome number, so such kind of the differentiation and the division has to be from another cell division, which is through mitosis. So this is how we can be understanding about the events which are taking place inside the Microsporangia when the anther is maturing, right? So we have MMC giving rise to microspores, and today we are going to be having more of the focus on understanding of the process up till here. So the formation of microspores, haploid microspores from MMC, right, from the microspore mother cell through meiosis. This entire process is the process which is called as Microsporogenesis. So we have the generation of haploid microspores from MMC, or the microspore mother cells, through the meiosis. Okay, and this process is referred to as microsporogenesis. Finally, let's try to understand the process of microsporogenesis. So as I said earlier, microsporogenesis is the process through which we have the formation of microspores, the haploid microspores, from a diploid microspore mother cell through meiosis in Microsporangia. Right? Now, let's try to get into the details of how this process is completed. So to understand this, let me give or take an example of a single microspore mother cell. So let's think that of the many cells inside the Microsporangia, so we have a single microspore mother cell. So let's assume that we have a single microspore mother cell, which is a diploid cell, because angiosperms are diploid plants, and because of which the cells would be diploid cell. So the diploid one of the microspore mother cell would then undergo meiosis to produce microspores. But since we understand that meiosis is consisting of two of the cell divisions, right? So meiosis has meiosis 1 and meiosis 2. Now this microspore mother cell would first complete meiosis 1. And on completing meiosis 1, it would be resulting in the production of two daughter cells. And these two daughter cells are usually enclosed in a membrane. So let's say the daughter cells are enclosed in a membranous body, right? And this membrane is actually known as callose membrane, right? It is a polysaccharide. So a callose membrane encloses the two daughter cells obtained from an MMC by the first meiosis. And these two daughter cells hence are collectively known as microspore diet. Diet means it has two cells, alright? So, and a callose containing two of the daughter cells is microspore diet. Now then, next the microspore diet, each of the microspore then would be undergoing, daughter cell would be undergoing and completing the second meiosis. So on completing the second meiosis, now we get 4 daughter cells, right? And those 4 daughter cells are again going to be enclosed inside the callose membrane. So, second meiosis forms 4 daughter cells, and these 4 daughter cells, which are known as microspores, are enclosed in the membranous body, right? And because of which such daughter cells are collectively known as microspore tetrad. So tetra refers to 4, right? So because we have 4 of the cells here, they are collectively called as microspore tetrad. And these microspore tetrads are all haploid cells, okay? Because the division that is taking place in the MMC is meiosis. So because of which microspores are haploid cells obtained through the process of meiosis from a diploid microspore mother cell. So each microspore mother cell would produce 4 of the haploid microspores. And these 4 haploid microspores, which are enclosed in the callose membrane, would eventually be would eventually be becoming unicellular microspores. And this happens when we have the breakdown of the callose membrane. So with the activity of an enzyme that is produced from the tapetum, and this enzyme is known as callase. So as we know, ase is a common suffix for referring to the enzyme. So callase is the enzyme which breaks down the membrane that is callose and thereby produces unicellular microspores. Okay, so we get now the microspores independently, right? So these are the unicellular microspores, which are all haploid cells, right, after the breakdown of the membrane, which is callose, right? And such unicellular microspores would then eventually be developing, they would be dividing and differentiating into yet another very important structure of the male body, which is the pollen grain, right? So which we are going to be taking up in our next tutorial. So now, what are these unicellular microspores? These unicellular microspores are the male spores, the haploid male spores, and they are all produced by the process of meiosis, right, inside the anther, particularly within the Microsporangia, right? And such kind of a process is commonly seen in vascular plants, plants which has the xylem and phloem, right? So vascular plants, so unicellular microspores are formed in some of the, you can say, pteridophytes, and in all of the gymnosperms and angiosperms, right? So these are the haploid male spores, which are the reason why we call them as microspores is because they have a comparatively smaller size as opposed to the female spore, right? So female spores are known as megaspores, whereas the male spores are called as microspores because they are much smaller, right? And male spores are produced through the process of meiosis inside the Microsporangia. And hence, if you can be relating this with another topic that we have learned in our previous year, which was relating with the alternation of generation. So we know that in plants they have two of the generations, sporophytic generation and gametophytic generation. So this is the understanding about the sporophytic generation, where a sporophyte is capable of producing spores, right? So the sporophyte body is the anther or the stamen, and inside of the anther, particularly in the Microsporangia, we have the formation of the spores, which are the male spores here, right? So the spores are called as microspores. And because the spores are produced through meiosis, unlike in animals we have the gametes produced through meiosis, but in plants we have the spores being produced by meiosis, and because of which the spores are going to be all haploid cells. And these male spores produced in the Microsporangia are known as microspores. So with this we come to the end of today's session. And I hope that you have understood with all of the major section of this tutorial. This is the assignment for you. You have 5 questions to be answered, and the questions are here. Now in the second part, I have a small activity for you, and for this you will need to make a brief visit to a nearest garden and observe any 3 flowers. Now in those 3 flowers, you will tell if the flowers are unisexual or bisexual. And while making your observation, don't forget to take a picture of the flower to be shared with me. Alright? So have a good time and enjoy the beauty of nature.